Student, today we will discuss about the another cell organelle and it is known as ribosome. Clear? And you know very well about the ribosome that it is non-membranous cell organelle and basically related with protein. This thing you had read already in your previous classes. Clear? So first of all, I want to tell you about what is the historical background of ribosome. Ribosome is first isolated by Claude by cytoplasm of bean roots. Okay, means he isolated it from cytoplasm, and that time he has given it as a name microsome. What name he has given? Microsome. Clear? But later on, in bean root plant, it was first properly reported by Robinson and Brown. Clear? Reported by Robinson and Brown. Robinson and Brown. Okay, so this is the information they have given, but they have not given it as a ribosome name. Okay, so the ribosome name was given by and first observed by in a particle form, and the name of scientist is Pallade. Clear? In 1955. So ribosome name was given by Pallade in 1955. So it's about the historical background of ribosome. Clear? These are the dense granules which are present in cytoplasm also and in cell organelles also. Clear? They are present in many forms. We can say that if they are present in cytoplasm directly, that time they are known as the free ribosome. If they are present in ribosome, because we know that they are present in prokaryotic cell also, in eukaryotic cell also. Clear? Ribosome is a one type of cell organelle which is present in prokaryotic cell as well as in eukaryotic cell. Clear? So when it is present freely in cytoplasm, that time it's known as the free ribosome. When it is present inside the mitochondria, that time it's known as the mitoribosome. Mitoribosomes. And when it is present inside the plastid, that time it is known as the plastidoribosome. Plastidoribosome. Clear? And one more form, when it is attached with any cell organelle like nucleus in a nuclear membrane in endoplasmic reticulum, that time it is known as the bound ribosome. Clear? So now only we have seen the four forms of ribosome in cell. When it is present in cytoplasm, that time it is known as free ribosome. When it is present inside the mitochondria, it is known as the mitoribosomes. When it is present in plastid, that time it is known as the plastidoribosome. And when it is present in attached form with any nuclear membrane or endoplasmic reticulum, that time it is known as the bound ribosomes. Clear? So after this we will read these are the forms where ribosomes are present. Clear? That is clear. Now I am going to tell you in which type of cells ribosomes are absent. Clear? So ribosomes are not present in mature sperm. In mature sperm, ribosomes are not present, and in mature mammalian RBCs. Clear? Mature mammalian RBC is also lack ribosome. Clear? So they too are the cell forms where ribosomes are not present. Clear? So after it I am going to tell you about the types of ribosomes. Ribosomes are of two types, 70S type of ribosome and 80S type of ribosome. Clear? 70S type of ribosome is basically found in prokaryotic cell and 80S type of ribosome is found in eukaryotic cell. Okay, first we will discuss about the 70S type of ribosome, which is basically present in prokaryotic cell as well as inside the mitochondria also. Clear? So here you can see the two units are present. One is known as the small unit and one is known as the large unit. Clear? So ribosome is made up of two units, large unit and small unit. Clear? So in large unit and small unit also the two components are here. Here 70S ribosome, 50S and 30S is present. 50S means large unit, 30S means small unit. Clear? If they live in a free condition, means when large unit is separate and small unit is separate, that time they are having the 50S and 30S form. Now you think that what is the meaning of this S? S is basically refers to Svedberg unit. Clear? Svedberg unit, because Svedberg only the scientist who has given Svedberg constant, also we use this term. Clear? This is one kind of sedimentation coefficient. So this Svedberg unit we use, so on the honor of him we use the unit as a S. Clear? So we are in large unit and small unit. Ribosome is always made up of two units, large and small. When they are in combined form, they form 70S. But when they separate out with each other, that time they live in 50S and 30S form. It's about the 70S ribosome. 70S ribosome, this 50 is also composed of 23S and 5S. Means 23S and 5S, they both are the component of large unit, and 16S is a component of 30S unit. Clear? As you can see here, 60 to 65% of this ribosome, clear, 60 to 65% of this ribosome is made up of rRNA. rRNA means ribonucleo RNAs. Okay, this is the rRNA. RNA is also three types: rRNA, mRNA and tRNA. You will read much more about it in 12th standard when you will read protein synthesis process. Okay, so here 60 to 65% of ribosome is composed of rRNA, or 35 to 40% of it, of 70S ribosome is composed of protein. Clear? So you can see here 34 protein and 21 proteins. Clear? 34 proteins are present in large unit and 21 proteins are present in small unit. So it's about the 70S ribosome which is present in prokaryotic cell. Now we will discuss about the eukaryotic cell. Like prokaryotic cell, it is also composed of two units, large unit and small unit. Large unit is made up of 60S and small unit is made up of 40S. If you combine them, you can say that ma'am it is of 100 is coming, but no. When they live in free form, that time they are having the 60 and 40 subunits. Clear? But when combinedly sedimented form, they are in 80S form. Clear? So 80S type of ribosomes are present in eukaryotic cell. Here also you can observe that rRNA and proteins are present. So here 45% is made up of rRNA, and in this ribosome 50% is this ribosome is made up of 50% of protein and 45% of rRNA. So you can see that the ratio is very close with each other. Clear? So in this case the large unit is made up of three parts: 5.8 sRNA, 28 sRNA or 5s RNA, and the smaller unit is having the 18s RNA. These are the composition of these large and small unit. Okay, so here you can see that the protein is also very important and rRNA part is also very important. So these are the types and formation of the ribosome. Now I am going to tell you the functions of ribosome. What is the main function? They are the sites for polypeptide. Polypeptide means protein synthesis. Clear? Peptide is a bond which is present between of two amino acids, and you know protein is a chain of amino acid. This is the peptide bond. Clear? And that's why the protein chain is also known as the polypeptide chain. Second thing, they provide enzyme and factor for the condensation of amino acids. Clear? For making this chain, we required how the amino acid will combine with each other. So it provide the platform for the combination of amino acids and for the regulation of the polypeptide chain. So I hope ribosome is clear. This is the most important, we can say, cell organelle, and if you have any query regarding this, just send me your question. Thank you, children.